Karyotyping has been used for many years, but what is a karyotype and why is it so important? Let's get started. All organisms have chromosomes in their body. These chromosomes are a structure composed of DNA as a very condensed form of chromatin. The complete DNA sequence in each cell of an organism is called a genome. Humans have 3 million base pairs which are found in the 46 chromosomes in the cells that form in our body. Chromosomes are made up of chromatin, and chromatin consists of DNA and protein. Human cells have 46 chromosomes, and these are organized into 23 pairs. Each pair has one chromosome from the biological mother and one from the biological father. One of these chromosome pairs is a sex chromosome. The sex chromosomes determine the gender of an individual. A genetic female has 2 X chromosomes, while genetic males have X and Y chromosomes. Since the mother can only give an X chromosome, the father determines the sex of an infant. The sex chromosomes are always counted as pairs, even though X and Y are not similar. The rest of the 22 pairs of chromosomes are considered as autosomes. Autosomes are any chromosomes that are not sex chromosomes. These 22 pairings are based on the sharing of similar characteristics. Therefore, they are called homologous chromosomes. This means that they are similar in features such as length and centromere location, but not identical to each other. As we already discussed, chromosomes contain the cell's DNA. Genes are parts of DNA that contain genetic information for inheritance of specific traits. Homologous chromosomes carry genes for the same traits. However, they can also carry different forms of the same gene. This is called an allele. This differs in specific traits such as straight hair versus curly hair. A karyotype is a photograph of pairs of homologous chromosomes. To prepare a karyotype, a cell sample is taken and is treated to stop cell division. Then it is stained so you can see each chromosome pair under a microscope. The chromosomes from a karyotype are typically taken from metaphase of mitosis since the chromosomes are condensed and thick. Now that we covered what karyotypes are, you might be wondering why is it important in the medical industry. This is because it helps doctors find out genetic disorders and missing chromosomes from specific locations. Now let's go into more detail about the specific genetic disorders doctors look for. This includes Downs, Edwards, Patos, Klinefelters, and Turner syndromes. By the way, if you're wondering, all these genetic diseases are incurable. Down syndrome is when a baby has an extra 21st chromosome. Some symptoms of this syndrome are how the baby looks and learns, loss of hearing, heart problems that are present at birth, and eye infections. Edwards syndrome is when a baby has an extra 18th chromosome. These babies usually have many problems and most don't live longer than a year. Heart defects, low set ears, slowed growth are some of the symptoms. Patos syndrome is when a baby has an extra 13th chromosome. These babies have a wide range of health problems, some of which are heart problems and severe mental disabilities. These babies won't live more than a year. Klinefelter syndrome is when a baby boy has an extra X chromosome. They may go through puberty at a slower pace and may not be able to have children. Turner syndrome is when a baby girl has a missing or damaged X chromosome. Some symptoms are heart problems, neck problems, and short height. And that is all about karyotypes. Thanks for watching.